So, hello everybody. Hi John, thank you very much for accepting the invitation to be here with us. Hello Gilberto, Paulo.
Hello.
Hi John.
Here.
Gilberto, hi John.
So today is our last section. Just before I give the words for Paulo to introduce John for us, please remember that today, in the end of the talk, we'll have the closing ceremony, which we will know who won the best seminar given by the students. And yeah, more information as well. And just again, remember to sign the list. We are going to give the link in a minute or by the end of this section. And yeah, also I'd like to thank once more our sponsor, which is Analytica, and everybody that's helping us in the organizing committee. And yeah, without further ado, I'd like to give the talk to Paulo, please.
So I'd like to begin by congratulating the My Friend is MASSA initiative and the Brazilian Proteomics Society for doing such a fantastic job in getting all these wonderful speakers to deliver these amazing talks. I couldn't be happier and more honored in introducing today's guest, Professor John Yates. John is the editor-in-chief of JPR, has an impressive H index of over 190, and several distinguished awards, among them the Beeman, the Thompson, the John Fenn Awards. John has consistently pioneered major contributions to protein science. Probably his most well-known contribution is an approach to correlate tandem mass spectral data of peptides with amino acid sequence in a protein database. Or simply put, we owe shotgun proteomics to him. In another vein, John has played a key role in the development of the Brazilian proteomics. His lab has consistently trained Brazilian PhD students and postdocs. He has actively participated in loco in Brazilian meetings, including those from the Brazilian Proteomics Society. I was thrilled with the opportunity to introduce John today, as I have known him for over 17 years. Being with John is always inspiring and a unique experience on learning the ways of the scientific career, especially on the importance of publishing. As he likes to say, if it ain't published, it doesn't exist. In being tenacious, as he says, just keep at it. If you have a bold idea that was rejected, the paper may be ahead of the curve. And in the importance of collaboration. Interestingly, he will also try to convince us that the best mass spectrometer out there is an LTQ. Did I get the last one right, John? Well,
No.
With no further ado, John, you have the word.
Ready?
Yep.
Okay. Thank you. Thank you for the invitation to talk. What I'd like to talk to you today about is some work that's been going on in my lab for almost as long as I've known Paulo. So it's in cystic fibrosis, and we're trying to understand how a single mutation causes this systemic disease, which is it's kind of surprising. It's just one single mutation that causes quite a lot of changes to a person's body. So let's see. So cystic fibrosis is one of the most common lethal genetic childhood diseases. It affects about 1 in 3,000 newborns in the EU, so the European Union. It's primarily a disease of Northern Europeans, but it's because as the Northern Europeans have spread throughout the world, that we see more and more of this disease in other places, Canada and the US in particular. So there's no cure available, but there have been new drugs which appeared in 2015 and 2018. One of the primary clinical phenotypes is accumulation of mucus obstructs airways and digestive tubes, and life expectancy is about 15 to 40 years. It's caused by a mutation of CFTR. This is the cystic fibrosis ion transport regulator, anion channel regulating chloride homeostasis, and deletion of delta F508, so phenylalanine position 508 in the protein, causes about 70 to 90% of CF cases. So what we've tried to do is to use mass spectrometry to understand misfolding diseases such as cystic fibrosis. When the gene was first discovered, it really wasn't recognized what kind of disease, what was causing the disease. But over time, as we've learned more about protein misfolding diseases, it's come to recognize that cystic fibrosis is almost a classic protein misfolding disease. And so we decided to explore this and try to understand the basis of the disease by looking at protein-protein interactions, post-translational modifications, and now we're trying to look more detail at the in vivo structure of the protein. For many years, this protein has been resistant to most methods to get at structural information for this protein. It's only recently in the last few years that as cryo-EM techniques have gotten better and better, that people have started to unravel at least the in vitro structure of this particular protein. So we're going to assess protein-protein interactions and post-translational modifications that play a key role in misfold CFTR recognition, quality control, and UPR induction. And then we're going to try to look at the direct identification of misfolding events in vivo as a way to better understand what might be going on. So first, I want to give you a little bit of background on protein folding. It's a very important process. As you know, proteins are not they don't exist as just a single monomer. They fold up into rather exquisite structures. And so as proteins are being translated, as the mRNA is being translated on the ribosome, the protein starts coming off the ribosome, and as it comes off the ribosome, it starts to fold. And it gets into some sort of folding intermediate, and this is being mediated by protein chaperones. It assumes some sort of folding intermediate, and that intermediate then folds into its native state. And once it reaches its native state, the protein is active and go off and do its job. So what can happen is that as the protein is being translated, it can result in stalled translation for a variety of reasons. And there's a ribosomal quality control system that comes in and will take the protein and assess whether it can be rescued or not. And if it can't, it gets degraded by the unfolding protein response system. So that stalled translation can also result in a misfolded or metastable intermediate. And that metastable intermediate can be attacked by chaperones to try to get it to fold into the native state. And if that doesn't happen, then the protein gets targeted to the UPR system, where it then gets eliminated from the cell. You can have mutations or proteotoxic stress, and proteotoxic stress could be something like a viral infection, which we all are quite familiar with right now, and that can cause proteins to misfold during this process, and you can again get these misfolded intermediates that need to be dealt with in some fashion. And these misfolded intermediates can also go on to form toxic structures, oligomers that can form amyloid fibrils, and there's a system to try to deal with those, which is called the autophagy system. So membrane protein biogenesis is very complicated. We have proteins that are getting translated into the ER. There's a quality control system in there. The UPR is in there, and so this is a complicated system. The proteins then head to the intermediate compartment, the ERGIC. And there's more quality control that's occurring, and as proteins are passing along this pathway, they're also being glycosylated, and then proteins will head through the Golgi where they get more complex glycosylation, and then eventually they get either inserted into the plasma membrane or they get secreted out of the cell. And this is such an important process, such a difficult process, that there's a network of about 700 different components that are involved in trying to get proteins to properly fold and to get them through this system. And membrane protein biogenesis is a highly complex process that is vulnerable to misfolding and aggregation. Most of the protein misfolding diseases that we're aware of occur because of some breakdown in this particular system, mutation in a protein or some other issue that might come up. So one of the questions that we're trying to ask by looking at cystic fibrosis, the cystic fibrosis protein, is how are misfolded proteins distinguished from correctly folded ones? There we go. And so protein misfolding is a cause of many common diseases. So there's these toxic gain of function diseases, which you're probably very familiar with: Alzheimer's, Parkinson's, amyotrophic lateral sclerosis. ALS is not very common, but it is a pretty lethal disease. I don't think anybody survives it. Famously, Stephen Hawking had a version of it that was very slow in its development, and he lived to be in his 70s, which is very rare. Transthyretin amyloidosis, and some cancers are caused by proteins that misfold and you result in a toxic gain of function. It can also result in loss of function, emphysema, cystic fibrosis, Fabry disease, Gaucher's, and again, there are cancers that have this as a hallmark. And so you can see that these can be involved in a variety of types of diseases, at least the ones that we're aware of. And so understanding the process can be very useful. Membrane and secretory pathway proteins are overrepresented in many of these misfolding diseases. So the delta F508 of the missing phenylalanine in position 508 in CFTR causes the protein to misfold. So it results in loss of channel function, prevents full maturation. The protein gets degraded by the ER-associated degradation pathway. This results in a loss of function. And so the protein is a complicated protein. This is the cryo-EM structure that finally came out in 2017. And you can see it has transmembrane domains, as well as it has a nucleotide binding domain 1 here and the nucleotide binding domain 2. And these help form the channel through which anions will pass through. So what was recognized, what was recognized fairly early on is that the gene was discovered in 1989. And by 1992, Denning discovered that this was a temperature-sensitive mutation, suggesting that it's suggesting that it was misfolding, although it wasn't fully understood at that point in time. But it was understood that if you decrease temperature from 37 degrees down to 30 degrees, the protein would fold enough that you would see the protein would mature by passing through the Golgi and the plasma membrane and getting more fully glycosylated. And so you would see a higher molecular weight version of the protein. And so this mature version of the protein gets labeled as, on a Western blot, gets labeled as band C. Band B is one that has passed partway through the system, and then band A is the immature form of the CFTR, it's ER localized. So the molecular mechanisms underlying this rescue have remained poorly understood, although growing the protein at permissive temperature has been sort of the gold standard for rescuing of the delta F508 protein. So trying to understand that process can be is useful for trying to understand the folding process in and of itself. So one of the first questions which we tried to ask are there differential interactions which are responsible for delta F508 misfunction? So we wanted to compare protein-protein interactions between the healthy protein and the disease protein to try to understand if there is a set of proteins that are involved in making decisions about the quality control. This is the process that we set up. It uses shotgun proteomics. We do a co-IP, so immune precipitation of CFTR, triplet digestion, and then we would use tandem mass spectrometry as a way to identify the proteins. And then we have a lot of software tools that we developed and used to analyze that data. And then we try to put the networks back together again to try to look at the differential interactomes. So we use shotgun proteomics, which I'm sure many of you are probably familiar with, is where you just take a protein mixture, you digest it up into peptides, you then introduce those peptides into the mass spectrometer via HPLC. For most of the work that we did, we have done LC-LC on these samples because that's simply the way we started. Nowadays, we probably wouldn't do it that way. And then we do automated tandem mass spectrometry. These data is searched with database searching algorithms, and then it's filtered and assembled into a list of proteins. This is a fairly well-established workflow and process, and almost all commercial mass spectrometers are capable of doing things of this nature. And now we are doing moving towards this is ProLucid is our third or fourth generation sequest algorithm. It's now been put onto a GPU card and Bruker bought the assets of a company that we had spun out of the lab called IPA, and which had created this tool called Pacer, which uses GPUs as a way to do real-time search. So we looked at this is the wild-type protein-protein interactome. It's all broken down by gene ontologies. So the folding pathways, degradation ER quality control pathways, trafficking, transport, endocytosis, with CFTR in the middle. And so we found that there are 368 interactors which are core, and 62 which are specific to wild-type, to the wild-type protein. Well, you may think that this is a lot of interactors that would be interacting with CFTR. The pull downs were done promiscuously, and so these are protein complexes which are interacting with CFTR. So there may be multiple proteins in a protein complex. This shouldn't think that this indicates that each and every one of these proteins is touching CFTR in some fashion. So when we look at the delta F508 CFTR interactions in the lung, we find that there are 368 interactors which are core with the wild type, but there are 209 which are specific to delta F508. And so within this 209, we surmise that there must be particular proteins which are making decisions about the folding status of delta F CFTR. And there are certainly lots of suspects that we can look at. The degradation ER quality control system or pathway certainly exploded in terms of the number of proteins that are involved and so forth. And so we have to figure out how to prioritize which proteins to follow up on. Now, here's another way to look at the data. So this is just a graph showing wild type only interactions at the top, delta F508 interactions only at the bottom. As I said, there are about 209 which are unique to delta F508 only, and 62 which are unique to wild type only. But there are also a bunch of proteins which have differential interactions between them. So they may be more abundant in delta F508 than they are in the wild type protein. And so again, these are organized by gene ontologies, ER quality control, degradation, folding, trafficking, and then plasma membrane. Plasma membrane is a section of this interactome that we have not explored very much, but I think there's probably some interesting biology going on in terms of those interactions. So how do we prioritize the important interactions? So comparing wild type and mutant provides insights, but not enough to prioritize the 200 or so differential interactions that were found. Which interactions are most important for rescue? So we had to come up with some experiments to try to figure out which ones of these are most likely to be interesting. And so we did that by taking the delta F508 again, and now we can rescue it by lowering the temperature of cell culture, lower the temperature down to 27, 30 degrees. It was also established by Bill Walsh's laboratory that if you use Hdac inhibitors, they will also rescue delta F508. And so we can look at the disease interactome against the rescued interactome, against the healthy interactome, and see what sorts of things are changing as we rescue the mutant form of the protein. And so here's the temperature shift experiment. It's shown as heat maps. These are the delta F508 interactors. These are the wild type interactors. You can see these are the ones that are unique to delta F508. These are unique to the wild type. So after 1 hour, we start seeing some changes. After 6 hours, we see some more changes. And then after 24 hours, we start seeing that a lot of the delta F508 interactors have gone away. And so this helps us prioritize which of those interactions might be more important. We also did the treatments with Hdac inhibitors. So these are inhibitors that inhibit 8-histone deacetylase enzymes. In the Walsh paper, he suspected that these inhibitors were attacking Hdac C7 primarily. So we also included an siRNA experiment where we knocked down Hdac C7. And this is Hdac inhibitor TSA, here's SAHA, versus the siRNA knockdown. And you can see that there are slight differences in terms of the interactomes that are being produced. SAHA has 211 interactors, which are unique to it. And so here's another heat map showing the different interactomes. Here's the delta F508 untreated. Here's the wild type untreated. And then after treatment with SAHA and then TSA, you can clearly see that TSA has a big effect. This is another drug that we tested for A, and then there's Hdac C7. You can see that it gives a different pattern than you see from some of these other patterns. So what we came up with is based on all this data, we were able to narrow down that 200 or so interactors down to 52. And then we tested each of those using an RNAi to knock down those proteins and see whether or not we could rescue. And so 48 influenced CFTR maturation in some form or another, and these interact with about 7 different subnetworks within the cell. So while you can't read this very well to see how these ratios are changing, here's it organized by the interactors organized by location within the cell, and also some cytofluorescence experiments that we did in order to show that these particular proteins were in fact co-localizing with CFTR at these various locations. So that's just some of the validation data. We also validated by doing a reverse IP. So PTP LAD1 interacts with CFTR when you pull down CFTR. And so we did a reverse pull down where we pulled down PTP LAD1 with an antibody against it, using that as the bait. And this is the interactome that we picked up when we did that. CFTR, delta F508 CFTR was part of that interactome. So it's verified that these two proteins were interacting as we had predicted in the original pull downs in the ER environment. PTP-LAD1 is known to be a ER resident protein. Okay, so now we want to test and see which of these targets, if any of these targets, has any therapeutic potential. And so we do this experiment, which is rather a crude electrophysiological experiment, where we can take primary CF patient cells or a cell line, CFBE cell line, and we can infect with lentiviral shRNA to knock down a particular protein. We then let these cells mature. It takes about 28 to 30 days, and they form a monolayer at a liquid-air interface. This is as they would in the lung. So this is a well-known process. And so once they form that, we can use this thing called an Leucine chamber, which we can then measure current across these cells. So if the CFTR is functioning properly, it should result in a passage of current through the system as the anions move across the cell membrane. So we should be able to measure that, and it'll tell us whether or not this works or not. So we're going to measure 8 novel interactors that we think are some of the best cases for this. Here's a control. Here's the temperature shift assay, the 28 degree, which is kind of a gold standard for rescue. And what you see is that when you treat with forskolin, which is a stimulator, you see that the current starts going up. And then you treat with genistein, which is a potentiator, and then you see the current really goes up. And then to make sure that what we're opening up is only CFTR, you now treat with a CF inhibitor, and that should shut down the current. And in fact, it does in this particular experiment. And so here's another one where we've knocked down this protein YBX1, and you can see that we're getting current. We knocked down TRIM21, we're getting lots of current. PABPC1, again, current. And then we test this legal S3BP, and we get no current. So this one doesn't work at all. So we looked at CFBE cells, which is the cell line that we did the interactome in, and almost all of them, with the exception of the legal S3BP, give us signals when we knock them down. And then when we looked at CF patient bronchial epithelial cells, we actually get better signal than we do in this cell line, suggesting that these are potential targets that one should look at in terms of therapeutics. Okay, so now we want to know about PTMs. So it was clear from the interactome data that we looked at that there are kinases and phosphatases that are involved in the interactome, and so we wanted to know what's the role of post-stational modifications in regulating this process. So we repeated a lot of our experiments. This time we made sure we had phosphatase inhibitors in there, and we did our analysis a little bit differently. Rather than just doing a straight up triplet digestion, we did a process we call triple digestion, where we used 3 different proteases as a way to optimize sequence coverage. And then we do a little bit differently with our database searching to try to cover more of the sequence and to try to look for unknown modifications that might be present, this blind PTM search. And then we do spectral validation, statistical analysis, and so forth. So this is the Cftr sequence, and this strategy results in about 80% sequence coverage. The regions that we might not expect to cover would be transmembrane domains, which you can see in some of this brown area. Those are transmembrane domains that we might not cover because they're stuck in the lipid bilayer. And there are also glycosylation sites, which we might not see sequenced because we didn't remove the glycosylation when we did these experiments. And so some of those regions might be ones that where we're not getting any sequence coverage. So to make a long story short, this is what we see. These are the modifications on delta F508 on the top part, and these are the modifications which are consistent with the wild that are in the wild type protein on the bottom part. We identified about 40 different PTM sites, and approximately 70% of them were new. There are 21 phospho sites, 6 methylation sites, and 11 ubiquination sites. You would expect to see higher levels of ubiquination on delta F508 because it's going to be targeted for destruction. And in fact, the pink sites are sites of ubiquination in the delta F508. We don't see much down in the wild type. So we found one site down here. We did find a lot of phosphorylation, and we found in some cases that modifications cluster in what we referred to as modification hotspots, which overlap with specific types of sequence motifs. We also looked at a lot of the sites that we found modifications and looked to see if there were any known mutations at those sites. And in fact, there were. The ones that are in boxes. The red ones are known mutations at those sites are known to create severe CF, and the ones that are in purple create a mild form of CF. So we looked at again. We did some experiments to more to quantify these sites with a little more certainty. So we did a few different kinds of experiments where we did a pull down using stable isotope labeling of heavy and light amino acids, where we did a pull down and then looked at the identification. We then did quantitation using stable isotope labeled peptides, which is called targeted mass spec, and we also did a TMT experiment doing this. So we quantitated these things pretty thoroughly with 3 different methods. And what we found is that there are phosphorylation sites and methylation sites that are important in the protein structure. So there's the TMT, the results we got in the TMT. But what we found is that these particular phosphorylation sites have a casein kinase 2 alpha 1 consensus phosphorylation motif. So this is the targeted mass spectrometry experiment where we quantified with phosphopeptides with heavy spiked in, and the wild type protein. We find that there is high levels of phosphorylation at T421, S422, and S427. And we find, so we looked at just the single sites, we looked at double sites, we looked at triple sites with all these different peptides, and showing you single sites for now, to see are these phosphorylated at all the sites, 2 of the sites, or just one of the sites. And the phosphorylation just has to exist at one of the sites at 421, 422, or 427, and not at 2 or 3. We then tested wild type by inhibiting the casein kinase 2A, thinking that was in fact to show that was in fact the kinase that was phosphorylating, and we knocked down phosphorylation significantly. And then at delta F508, we find there are very low levels of phosphorylation at any of these sites. So we did some more experiments. This is a Leucine chamber experiment where we took delta 508 and we put it at the permissive temperature of 28 degrees, and that should cause it to recover and to function. DMSO is just the carrier for the drug, and you can see that it gives us a fairly decent signal. It's also known to permeabilize cells, which may be why it does that. This DMSO may not be a great control for these experiments, but when we use CX495, we knock down the current that we see coming out of delta 508 at 28 degrees. So we also did a knockdown experiment on the casein kinase, and that prevents rescue of delta F508 channel function as well. So that's the shRNA right there. So we analyzed the PTM landscape across using different type of drug treatments known to partially restore delta F508 CFTR function. So 57 sites across 12 different conditions, and we do find that this particular site at 421, 422, 427 is a particularly important site within the protein. And in fact, we then compared to several other mutations which are known. So there are some mutations which are called trafficking competent. So this G551D protein is it gets out to the cell surface, it just doesn't function. And then there's the R117H, gets out to the cell surface but doesn't function. And then trafficking impaired would be the delta F508, and there's another known mutation, N1303K, which doesn't traffic out of the ER as well, misfolds. And so you can see that phosphorylation in the wild type is fairly high, phosphorylation in the G551D is high, and then the R117H is also phosphorylated at these sites, although not as much. And then the delta F508 and the N1303K phosphorylation is very low, if at all present. So we think there's a PTM code for CFTR maturation. So we think that if you have phosphorylation at one of these three sites plus methylation, you get full maturation. And if you don't, so if we take wild type CFTR and we knock down the kinase, we don't find much phosphorylation at any of those sites, but we do find elevated levels of ubiquitination blanketing that site. That results in degradation. Same thing with the misfolded delta F508 CFTRs. This is just the misfolded normal protein. And then if we rescue delta F508 CFTR at 30 degrees, we restore phosphorylation and methylation. We get full maturation. And then if we take the misfolded delta F508 at 30 degrees C, which should be the permissive temperature, and we knock down the kinase, we again see low levels of phosphorylation and ubiquitination blanking those sites. So we think this represents a PTM code for when the protein has matured. So why might that be important? So we had done some experiments earlier on, but we didn't have this PTM information to try to interpret this in a different way. What we were simply looking at, we were doing cross-linking mass spec to look at how the wild type NBD1 domain interacts with the HSP90. And this was the in vitro experiment just done in the test tube. And the wild type NBD1 domain is not phosphorylated since it was produced in insect cells or E. coli or something. So it wasn't properly phosphorylated on that site. And what we found is that HSP90 binds, when we cross-link, binds at this 421 site in vitro. So we then went back and we looked at some of our, we did another experiment, IP experiment, where we looked at the wild type with the inhibitor, the kinase inhibitor versus the wild type. And what we found is that HSP90 alpha and beta subunits quadruple in the amount of protein that's binding to the to the NBD1 domain when we do these experiments, suggesting that that's an important site where HSP90 can bind, and once it binds there, it doesn't let go, and the protein gets targeted for destruction. Phosphorylation defects also trigger ER quality control failure. So if we then treat the wild type protein with the kinase inhibitor, we see that the wild type interactome gains 59 of 71 interactors normally specific for delta F508 CFTR, and that mediate ER quality control and degradation. So suggesting that the phosphorylation is very important. So now we want to know, we were curious, what are the folding signatures that are being produced in this system that might be signaling to these interactors. And there are a number of ways that we look at a structure. A lot of these are structures that are done in vitro, but increasingly there are methods that are coming along to look at things in vivo. So methods that you know about, such as NMR, ion mobility, native mass spec, cryo-EM, can look at single proteins or protein complexes increasingly, X-ray crystallography. But there are these methods like FPOP, and FPOP is a fast photooxidation process where you can put OH labels on the outside the solvent accessible regions of a protein. SPROX is fairly similar, and we've been developing a strategy which we call covalent protein painting, which is an in vivo method. And this is a method where we can do this in intact whole cells as well as animals, because it's relatively conventional chemistry that you are probably very familiar with, that we've just exploited for this particular purpose. So these are collectively called protein footprinting methods to gain more conformational information. So this is a hot area right now, the 3D proteome. People are using a variety of methods to try to get at that information. So the hydroxyl radical strategy, the FPOP method, is used by Mark Chance, Mike Gross, and Lisa Jones is trying to take this strategy more global to do whole proteomes. People have also used carbines. Mike Fitzgerald's lab has done oxidation, method he calls SPROX. He uses hydrogen peroxide to do this, but without the use of a laser. This method, FPOP, uses a laser to trigger the breakup of hydrogen peroxide. Hydrogen deuterium exchange has been around for quite some time. I think Walter Englander gets credit for having discovered it. Pat Griffin, my classmate from grad school, is very good at doing this. Fluorophore, you can do fluorophore interactions, and molecular painting method that Lance Leota has developed. And Richard Vachet's lab has done a lot of chemical labeling type strategies, including ethyl pyrocarbonate and other methods. And there's a protease sensitivity method, which has been developed by Paula Picotti's lab at the ETH, where they do fast proteinase K digestion followed by trypsin digestion as a way to look at how things might be different within a cell. So the strategy that we developed, we started calling it molecular painting. We've now reverted to covalent protein painting, since molecular painting was already taken by Lance Leota, but covalent protein painting may be taken by somebody else. So we're just calling it that for the time being, I guess. And so what you do is you can take your intact cells or your intact tissue and you label it with a light isotope version of formaldehyde. And you're all probably fairly familiar with fixing cells and so forth. With formaldehyde, if you let it sit long enough, it cross-links and it fixes tissue in place. And it does that by forming a Schiff's base at lysine residues. And this is a strategy that's been used in proteomics for a while, where you can then reduce the Schiff's base to using cyanoborohydride, where it then forms a dimethyl tag. And if you do that with different weights, formaldehyde and cyanoborohydride, you can introduce stable isotope labels into this. And so what we will do in our first labeling experiment is we label with light isotopes. We put a light dimethyl tag at lysine residues, but it only interacts with or labels those lysine residues which are surface accessible. We then digest. We typically will do this with chymotrypsin because we are labeling lysine residues and we want to be able to label them. And if we digest at this point things which we have not labeled, we will get sort of erroneous results where we won't be able to match things up. So we then digest, and then we introduced heavy isotope labeling, where we use heavy formaldehyde and heavy cyanoborohydride, and we can introduce a heavier version of the dimethyl tag. We then run that through the mass spectrometer, and we identify and we get differences in peak heights for peptides, which tell us how surface accessible a particular lysine residue was. So we can quantify based on those differences, and this gives us information about solvent accessibility versus solvent exclusion. And we've developed some software tools to try to interpret this data. So, for example, here's CFTR, and what we've done here in this table is we've taken our covalent protein painting mass spectrometry, and we've looked at lysine residues, which our data using CPP suggests it should be accessible, versus the cryo-EM structure, which came out in 2017, being accessible. So this ICL2, K273, our data for the wild type suggests that it's less than 10% accessible, but greater than 95% accessible for Delta 508. and the wild type cryo-EM structure says it's inaccessible. So it says it fits very well with this measurement that we got for the wild type protein. And then you can see for some of these other particular regions, so NBD1, NBD2, in the R region, where we didn't get any data from the cryo-EM. We couldn't interpret any data from the cryo-EM data. So overall, it looks like it's pretty consistent with what the cryo-EM sequence is giving us. So here's a Cpp experiment where we just took delta 508 and wild type CFTR. We went through and repeated the experiment 4 times. This is light, heavy, and these are different domains within the protein ICL2, NBD1 domain, R region ICL4, NBD2 domain, and so forth. And the dark blue is the wild type protein, and the red is the delta 508. So one thing that you should see that's very different right here is that K273 in the wild type protein has very low accessibility, whereas in the delta 508 protein, it's got very high accessibility. So we then did the label swap experiment, heavy, light, and we see pretty much the same results when we do that experiment. So we see conformational differences are located in the transmembrane helix 1, ICL2, NBD1, and NBD2 domain. So here's the K273 MS1 scans, and you can see in the wild type, you can see that it's mostly inaccessible. So yeah, mostly inaccessible. And then delta 508, you see it's mostly accessible. And what's unique about this K273 domain is that it is thought to be a ball and socket, have a ball and socket type relationship with NBD2 domain. And so it's forming this this bridge here, and the K273 sits inside the socket, and so it should be inaccessible in the wild type version of the protein. And what we see is that for the 2 trafficking incompetent mutations of CFTR, we see that K273 is accessible, suggesting that it has a more open structure at that particular site. So we've done a bunch of different experiments where we're trying to look at using drug treatments and so forth to look at how that accessibility of 273 might change. So at 28 degrees C in delta 508, where we would see it to be more wild type like, we do see that we improve on the accessibility or the inaccessibility of K273. It drops down to 88.9 from just the standard 95.04. If we treat with VX809, which is one of the drugs that's approved that's on the market for CFTR for CF patients, we see that it really doesn't change the accessibility very much for the delta 508. If we now treat with VX770, which is one of the other drugs that's approved from Vertex, and what this drug does is it causes the channel to open up and stay open, and that this is primarily used to treat G551D mutation, which can get out to the cell surface, but the channel won't open. This opens the channel and leaves it open, and what you can see is that the solvent accessibility is now it's just now fully open. Whereas in the normal wild type, you see it's about 60% or so accessible. So coupling of ICL2 to NBD2 upon activation in vivo. Uncoupled state is an inactive state occurring in vivo that was not captured by cryo-EM or homology. And so you can see here's some structures that have been done. More recent paper, 2020, looking at the dephosphorylated cryo-EM version, the phosphorylated cryo-EM version. So you can see the NBD1 domain and NBD2 domains are separated, and then they're pulled together in the phosphorylated version. And then this is an open configuration 3D model for it. So we looked at, we mutated that site, that K273. We converted it to an alanine, and we find that changes the solvent accessibility and charge of ICL2. It alters CFTR biogenesis and leads to accumulation in the TGN endosomes. So if we take the wild type protein, we convert that K273 to alanine. It now starts acting as if it's delta F508 version of the protein. So we've been trying to build a model of CFTR in the inactive conformation. So looking at K273 buried, partial K273 exposure, and then full K273 exposure to try to understand how this interaction between K273 and the NBD1 domain may be affecting the ability of the protein to mature, so that we can then start thinking about how we can fit what we have to do in order to fix it. Closer look at 273, and there's an electrostatic interaction with P960 and M961. We're trying to do some modeling on this to get this to work, but we're struggling to get people that really know how to use Rosetta to help us with this particular problem. And so we've been Pat Griffin's lab has been working with us. They know a little bit about how to use Rosetta, but they're not real experts at it. So we're using that to try to get at some information for this. And so here's where we've gotten so far with our model for this. You can see how that, so it shows ICL2 unfolding with the NBD2 displacement. That's about as far as we've gotten so far with this. So this conformational change of ICL2 upon CFTR activation is accompanied by changes in the ion permeation pathway. We think that the channel is supposed to be closed as the protein is moving through the ER pathway to prevent anions and so forth from passing through the channel, and that when it's found not to be the case, it's thing is targeted for destruction. So that's one of the hypotheses we're working on. And this CPP method is helping us to try to understand this. So we're able to look at wild type treated with VX770, which gives us this, and then comparing that to delta F508. So phosphorylation and methylation, just to summarize, so phosphorylation and methylation sites in the regulatory interface element define a minimal PTM code for CFTR maturation. Is phosphorylation and methylation prevented by misfolding? So that's, why is it that it doesn't get phosphorylated or methylated at those sites in the immature protein? The PTM code is clearly disturbed in CF and contributes to the disease. And then does this PTM code define a maturation code for other membrane proteins? We've got some evidence to suggest that there is a class of membrane proteins where this PTM code does in fact exist in some form or another. If we do a sequence search for it, it's not particularly clear that code exists in all sorts of proteins. But if we do an experiment where we block phosphorylation, we do see fairly dramatic differences in the number of membrane proteins which are maturing. We're also working on trying to get the structural model for what might be happening in delta F508. Make drugs for this to see if we can use the knowledge that we've gained to try to fix it. Software support has come from Salvador and Diego. Diego, for initially when he was around, he's now been at Qualcomm for probably 7 or 8 years. And Salvador has done a lot of work. We've had funding from the NIH as well as Cystic Fibrosis Foundation over the years. And there's the rest of the lab. And so here's my, Fabio is my only current Brazilian in the lab. Yeah. So that's Fabio, and he's been working on top down. And thank you very much. Happy to answer any questions you might have.
Thank you very much, John, for this highly interesting talk. And we can see that you're doing like a hell bunch of things to validate and validate. I know that this is now the message that you start saying in JPR that we should not just do proteomics, like we really need to validate a lot, and that's what you've shown to us.
Right. The proteomics really should be giving you hypotheses to start
exactly
testing.
Exactly. It's a hypothesis generating method, right? And then
Correct.
you need to keep digging to answer more deep questions. We do have a lot of questions from the audience. I don't know if I put here, can you read it, or you prefer me to read for you?
I can see them.
Okay.
So the question is, how do you deal with proteoforms in your PTM analysis? Really great question. Right now, we get fairly comprehensive identification of modifications. What we can't tell is patterns, and that's what people refer to as proteoforms. So there might be one protein which may have slight variations of patterns of modifications. We think we've done a pretty good job of dealing with the proteoforms that might be present at the particular sites in our PTM code. But those are all fairly close together, so that was kind of easy to do. But right now we are not dealing with proteoforms on the protein level.
Mm-hmm. If I can also make a question more or less about it regarding your method to see the accessibility of the protein, right? That you do dimethyl labeling. If I understood correctly, you do it before lysing the cell, right? Because your protein is it's a channel, right? So
right.
you make the labeling in your cell culture. Is it correct?
Correct.
Okay, and how is it? Does the dimethyl labeling go inside the cell too, or you're going to label only the outside part of your channel?
No, so formaldehyde. So people who are biologists, cell biologists, will be really familiar with the use of formaldehyde. That's how you fix cells. And histologists and physicians, when they're preparing cells for microscopy, they're fixing the cells. They use formaldehyde, and so it penetrates through tissues. It penetrates through cells quite readily, and it's the reaction's very fast.
Okay, yeah, very interesting. So Magnum has a question, some questions for you actually.
So this is, I mean, there's probably more to this question than meets the eye, because PTMs are often what we call one-hit wonders. So those of you who are from the Rio area, the girl from Ipanema was probably characterized that as a one-hit wonder for that particular band. They never had a hit of the same caliber, and so frequently we have to, if they're really important modifications, we will validate them by hand. So we will go in, look at the spectrum, and make sure they're high-quality hits. But generally speaking, the spectra need to be very high-quality spectra, very good spectra, and the matches need to be very good matches, because those can be particularly tricky things to validate. They can be done automatically. There are some software programs that people have been developing, particularly for phosphorylation, that try to make an effort to validate. But if you have a modification which is going to redefine biology forever, you want to check it manually and make sure it's a good match.
Yeah. He has another question on top of that.
Yeah. So I'm not sure that de novo sequencing is robust enough yet to help with that. There are other algorithms which can help with assessing the match. Even doing some simple things like taking a different database searching program and researching the data and make sure it matches up to that modification site would be useful. For phosphorylation, there are experiments you could do where you could take the fraction that contains your phospholipid of interest and you could treat it with phosphatase to remove it and make sure you see, the peptide without the phosphate on it.
Okay. Let me ask you about the workflow that you use to study the ubiquitination. Is it all antibody based? You do a pull down of the ubiquitination?
No.
Oh.
No. We were so we have a strategy that we use, which we call triple digestion. And so we will take a sample, divide it into 3 aliquots, treat it with trypsin, subtolysine, and elastase. And so these 2 last proteases are non-specific proteases. And so we do a time-limited digestion with that. And then we can either recombine them or run them all separately. Depends on how complicated the mixture is. And then when we do that, we get very high sequence coverage across the protein. And we also get ladders of sequences. This is another thing that helps with validating modification sites is that, and you can go and look at we had the 2002 paper on this. Mike McCoss was the first author on it. But what you get is a ladder of different sized peptides. And if they're in the same region, and they will confirm help confirm the modification site.
Okay.
So it helps us to identify these sites. What it does lack, depending on the complexity of the mixture, it can lack dynamic range.
Okay. So then by doing that, you can distinguish between mono and polyubiquitination.
No, we're just looking for the digly tag.
Okay.
So we can't tell if there's a ubiquitination chain on there or not.
Because it does change, right? I think it's for degradation, it needs to be polyubiquitination, while for just signaling, you can have just mono ubiquitination. Right? I'm not very familiar with that, but I know it. I think there is this difference. And it's
you're right. There is a difference, and we're missing that information.
Okay.
I mean, since it's being degraded, we assume that it's getting polyubiquitinated.
Yeah, exactly. But it's, it was quite interesting that, as you've shown, that there are not that many ubiquitination sites. You would expect to have more, right? Because you say that it's being unfolded, so it's probably going to be degraded. And as you've shown, not that many. But on the other hand, a lot of fossil. And then for fossil, you're using titanium dioxide, or
no? We're just doing that straight up the same way we're using our triple digestion method. Okay.
We have more questions here. Marcia, that I know that she was in your lab a while ago too.
Yes, you can use that same strategy to study other diseases. In fact, we have been looking at other diseases and looking at the protein-protein interactions and so forth to try to better understand them. I think it's a good way to do it because it's very targeted, and the information that comes out of it is very actionable.
Mm-hmm.
When you do a protein expression analysis, it's kind of hard to follow up on that data because you can see a lot of things changing. And they may not be the follow-up may not be as logical.
Okay. How long it takes, John, to take the whole set of workflow that you've done? I mean, if you think I think cystic fibrosis is, I don't know if it's something that it's more personal, it's individual, this changes. Because my question is, if you want to do more or less this workflow that you're doing for personalized medicine, do you think it's feasible? Like, take the sample and then try to sequence the protein, see the mutations, and then try to express this protein and test, for example, different drugs to see which one is more effective for that particular mutation. Do you think it's feasible, or it's just a lot of work that probably it's going to be very time consuming?
Yeah, I don't think it would be possible yet to try to use that as part of a personal medicine type strategy.
Okay. One more question here.
No, we haven't really thought that carefully about that. So this is, have you thought to investigate if there are differences in the glycan structure on CFTR between the wild type and the cystic fibrosis condition? Primarily because there will be big differences between the two, because the delta F508 protein doesn't exit the ER. And so most of the glycosylation is occurring in the Golgi. And so we see there'd be very little glycosylation on the delta F508 version of the protein. So what might be more interesting would be to take the delta F508 lower temperature in culture and then recover the mature protein and see what the glycosylation looks like versus the wild type protein. That could be a lot more interesting, because there should be more glycosylation. So we had done an experiment many years ago where we looked at I forget, we treated a rat with a particular chemical, and then we were looking at proteins, and one of the proteins out in the liver, one of the proteins that was the proteins that were upregulated, classes of proteins that were upregulated were glycosylation enzymes. And so the thinking was that the proteins were starting to unfold, misfold, and the glycosylation enzymes are being upregulated to try to help with the folding of those proteins. And so it is known that when that cells that are under proteotoxic stress, glycosylation patterns will be different.
Okay, I think that was it for most of the questions here. Gilberto.
Well, John, thank you very much for accepting our invitation.
Oh.
And today is the last seminar, and to have you presenting it is an honor for us. As Paulo said, Brazil owes a lot to you for your help in developing proteomics in Brazil. We hope we stay close together. You still keep receiving Brazilian students and helping us to go ahead and overpass this, the economic situation in Brazil, and helping us to spread the proteomics gospel around the country. Thank you, John.
My pleasure, and thank you for the invitation to talk. And hopefully we'll see each other in December for IMSC. Maybe?
We will. Yeah, we will, of course. We'll host you here.
Okay, thank you very much.
Yeah.
I'm gonna sign off.
Okay, thank you, John.
Thank you very
Bye.
much. It was really, really nice.
Thanks.
Bye.
Bye. Tá com você, Gilberto.
Tá comigo? Não, tá com o Fábio agora, né? O Fábio que entra agora.
Não, podemos começar, mas eu posso começar com a sessão, né, de premiação dos alunos, né? Só divulgar, né? Agradecer mais uma vez todos os alunos que submeteram o seu resumo, né, para avaliação pelo MASSA, BrProt, tá? Foram 17 alunos que submeteram o resumo, tá? Todos eles de excelente qualidade, como eu já havia falado. Todos eles foram avaliados por uma banca científica, né? Eu também gostaria de agradecer nominalmente a todos os membros dessa banca, né? O professor Carlos André, da UNB, o professor Francisco Campos, lá da UFC, né? A Luciana Pisati, que é professora da UFRJ, o Mário Sérgio Paulo, professor da Unesp, o Paulo Carvalho, né, que estava aqui, então, pesquisador da Fiocruz, e o professor William Castro Borges, da UFOP, né, de Ouro Preto. Então, eles, assim, foram excelentes em avaliar todos os resumos, todos eles comentaram, premia todo mundo, foram todos os trabalhos muito bons, excelentes, todos de excelente qualidade, mas 6 foram selecionados, né? Todos os 6 apresentaram, as apresentações também foram ótimas, e a mesma comissão participou da seleção dos trabalhos, ou do melhor trabalho, ou da melhor apresentação. Na verdade, todas elas foram excelentes apresentações, tá? Mas um deles foi selecionado. Bom, para os 6 alunos que apresentaram a ideia, né, a gente se reuniu, né, todos aqui, da comissão organizadora, os professores, e a gente resolveu fazer o seguinte: todos vão ganhar uma menção honrosa, né, um certificado de menção honrosa, né? Então já fizemos os certificados hoje, né, Tiago e Gilberto e Mário. Então a gente já tem um certificado de menção honrosa em agradecimento. E a gente também se reuniu, como todos os trabalhos eram bons, a gente imaginou como é que a gente poderia premiar esses alunos todos, né? Todos eles foram muito bons. Então, surgiu a ideia da gente, de patrocinar, né, a apresentação desses alunos na RIOPO 2021, que é uma RIOPO que vai ocorrer online, né, a RIOPO Reconnect 2021. Então, para todos os alunos, a ideia é patrocinar a inscrição deles, né? Então, a gente vai arcar com a inscrição na BrProt, aqui uma referência MASSA, com a inscrição desses alunos, dos 6 alunos que apresentaram. Claro, a ideia é que vocês se inscrevam, mas que apresentem um trabalho, né? Então, vocês podem inscrever o trabalho, fazer a inscrição no congresso, submeter o trabalho, e a gente vai cobrir as despesas com relação à inscrição de vocês, tá? A ideia é vocês fazerem isso, podem mandar para a gente a confirmação de inscrição e de submissão do abstract, a ideia é reembolsar vocês, tá? Quem tiver o interesse de participar da RIOPO Reconnect 2021, tá? Bom, e agora, sem mais delongas, né, a ideia agora é falar o trabalho que recebeu o maior número de votos, né? E, assim, o trabalho que... Deixa eu ler direitinho aqui, né, para não ter nenhum erro. Mas o trabalho selecionado pela comissão foi o estudo de repertório serológico de anticorpos de cavalo contra veneno de aranha do gênero Loxosceles, de importância médica no Brasil. Então, parabéns, Manuela, Cristina e Emiliano Ferreira, tá? Então, a ideia é a gente dar um certificado de premiação para você, tá? E também o prêmio de destaque, talvez, para você, a ideia é te passar um cheque científico, né? Seria isso, né, gente? O nome...
Não é cheque, não. Manuela, dá o número do Pix, a gente passar um Pix para você.
Agora é Pix científico, né? Moderno. Então, a ideia é fazer esse Pix científico. Então, a ideia é a gente passar para você um valor referente a qualquer custo que você queira comprar, um valor de R$500, né? Relacionado à sua pesquisa, ao estudo em proteômica. Então, a ideia é passar esse valor para você, tá? Então, parabéns, Manuela, parabéns a todo o grupo, né? É um trabalho em grupo. mas você realmente fez uma bela apresentação. E parabéns aos demais, né? Parabéns também aos outros alunos que, infelizmente, não puderam participar, né? Então, tiveram que ceder o lugar para outros alunos. Então, é isso, gente. Foi um prazer participar dessa seleção dos estudantes, dos melhores trabalhos. E parabéns, né? A gente se encontra aí no próximo ciclo de seminários.
E, Fabinho, se eu puder só complementar, já fica aí de incentivo para as pessoas que não enviaram trabalho nesse My Friends Massa, né? Já fica aí de incentivo, a gente já chama vocês para o próximo, todo mundo mandar um resumo. Quando a gente tiver mais informação, né, de quando vai ser, tudo a gente já passa para vocês, já fica aí o incentivo para vocês enviarem. Quem conseguiu agora, usa isso como um incentivo para mandar de novo. Quem sabe no próximo vocês vão ser, né, premiados com o primeiro lugar. Parabéns para os 6, parabéns para os 20, né, que o Fábio falou que enviaram. E é isso aí. 17. Se eu puder, então, só antes que a gente esqueça, eu não sei se o Gilberto ia falar isso, eu só queria agradecer demais, mais uma vez aqui, a comissão organizadora. Então, a Verônica, que é aluna do Giuseppe, lá da USP, o Luiz, que é aluna aí de vocês da UFRJ, a Jéssica também, que é da UFRJ, a Ana Carolina, que é aluna aqui da UFMG, e, obviamente, a gente também, né? O Magno, o Fábio, o Gilberto, o Giuseppe, eu. Foi um trabalho bom que a gente teve aí nesses últimos meses, né? Foi um prazer fazer e mais legal ainda ver que eu acho que foi um sucesso e que a ideia é a gente ir continuando, não?
Perfeito, Thiago. Foi excelente participar dessa equipe.
Eu queria dizer umas palavrinhas também. Posso, Thiago?
Lógico.
Obrigado. Hoje a gente fecha mais um ciclo de seminários, né? My Friends Massa da BrProt. Então, eu queria agradecer aos doutores Tiago Verano Braga, em nome da BrProt, né? Giuseppe Palmisano, Magno Junqueira e Fábio Nogueira, pela comissão organizadora dos seminários. A comissão julgadora também, que é um trabalho difícil, né? Muito difícil avaliar os pares, pelo belo trabalho que ela fez e pela serenidade que ela teve no processo todo. A todos os alunos, como o Thiago já agradeceu, né? Talvez eu esqueça alguém, mas que eu me lembro, Ana Carolina, Jéssica, Verônica, Felipe Luiz e outros que porventura tenham ajudado, nosso muito obrigado. Mas, principalmente, nada disso aconteceria se a gente não tivesse o outro lado, que é a audiência. Vocês superaram todas as nossas expectativas. A todos vocês que assistiram, acompanharam, e eu diria, se mantiveram fiéis, vindo semanalmente assistir os seminários oferecidos a vocês. E, obviamente, a nossa patrocinadora, a Nova Analítica, que sempre está presente nesse momento. Sempre que a BrProt precisa de patrocínio, seja ele pequeno, seja ele maior, a Nova Analítica está presente. Ela torna possível também a realização desses seminários. Então, a BrProt manda a todos, né, o nosso obrigado por nos ajudar a disseminar esse evangelho proteômico pelo país. Vocês todos, inseparavelmente, inegavelmente todos, mostraram que vale a pena fazer ciência. Boa ciência, eu diria, no Brasil. O grupo que organizou, que participou, não quero falar só de organização, mas que participou da organização em todas as áreas, né, do seminário, foi um time. Nós conseguimos montar um belíssimo time. Esse time não mediu o tempo, não mediu esforços, partilhou os seus saberes, partilhou os seus esforços para trazer a todos vocês o que cada um tinha de melhor para dar. Falta dar os parabéns também aos alunos selecionados, né? E a Manuela pelo melhor trabalho. Tenho certeza absoluta, pelo que eu ouvi, que vocês todos terão um belo futuro e um grande sucesso científico espera vocês. Outro ciclo, agora 2021-2, começa no segundo semestre. Minha sugestão, é que desde já vocês devem começar a afiar suas micropipetas, preparar suas máscaras para trabalhar no laboratório, aguçar sua criatividade e preparar os trabalhos para as novas apresentações que virão, as novas inscrições e apresentações. A hora é esta, quanto antes começar, melhor. Mais dados vai colher, melhor será o trabalho. Na realidade, foi pela ação de todos, né? Todos esses citados e os não citados, que me desculpe, que agiram por um bem comum. Todos criaram, todos sugeriram, todos se dedicaram e todos se comprometeram, além do que seria possível, né, para que a gente chegasse ao dia de hoje. Da minha parte, pessoal mesmo, só me resta dizer que eu tenho um orgulho enorme de todos vocês serem meus colegas e amigos. A recíproca é verdadeira. Eu tenho uma enorme alegria, um enorme prazer de acompanhar vocês, de termos seguido juntos nessa jornada da BrProt. Eu dou meus agradecimentos, meu obrigado a todos, e espero que vocês recebam minha admiração profissional, que é de todos vocês por direito de conquista. Eu amo vocês. Beijos. Era isso, Thiago.
Isso aí. Magno, você quer falar alguma coisa?
Vou comentar uma coisinha rápida, mas eu já fui muito bem representado aí pelos meus colegas. Acho que eu só queria dizer assim que é muito bonito ver como a espectrometria de massas e todo o campo da proteômica vem aprendendo a fazer perguntas biológicas cada vez mais importantes, né? Mais desafiadoras, né? Então, e isso está cada dia se expandindo mais, né? E eu acho que isso foi assim, a lição que a gente leva de todos esses seminários bonitos, né? Também, né? Além de outras coisas. E dizer uma coisa que acho que o Gilberto esqueceu de comentar, que a gente está também expandindo os nossos patrocinadores do BR, do My Friends Massa, né? Então, acho que outras empresas podem se agregar também e ajudar. Então, aí tem várias outras empresas que a gente pode citar no campo da espectrometria de massas também, né? Que podem nos ajudar, porque a gente não conseguiu fechar, por exemplo, os custos completamente dessa edição do My Friends Massa, então a gente precisa também desse suporte para resolver algumas questões técnicas, né? Aí por debaixo dos bastidores, que o Thiago sabe muito bem aí como é que funciona. Mas queria agradecer também os meus colegas, né? Foi ótimo. Interagir com eles é sempre maravilhoso, a gente troca muito, a gente aprende muito e faz ciência juntos, assim, discutindo sempre as questões que realmente nos excitam aí, né? que a gente vê que podem impactar a sociedade e trazer, responder à nossa ansiedade, conhecer um pouco o nosso universo, né? Mas é isso. Obrigado, pessoal. Foi ótimo.
Só um comentário rápido também, agradecer todos os palestrantes. E assim, né, que os palestrantes, como o Magno falou, o Gilberto, eles deixaram um pouquinho do conhecimento deles aqui com a gente, né? Então, agradecer a todos eles, principalmente os nacionais, né, que se desdobraram e dão excelentes apresentações, também os internacionais por casarem o fuso horário, né? E lembre-se, no próximo ciclo a gente já tem perspectivas de grandes seminários para o próximo ciclo, tá? Então a gente já pensou os seminários para o ano inteiro, né? Então a gente tem grandes perspectivas aí para o próximo seminário. Então fiquem atentos, tem um grupo do WhatsApp lá, a gente vai renovando as informações lá, tá? Então fiquem atentos que a gente vai ter aí mais novidades para vocês. Obrigado, pessoal.
Beleza. A data a gente ainda não sabe, né? Quando que vai ser, né? Vai ser, acho que era outubro. Tem que ser antes, né? A gente pensou ainda ser nesse ano ainda, só que
Isso.
um pouco antes, lembrando que a gente tem o internacional no final do ano, então a gente tinha pensado outubro, né? Mas aí fica aí a deixa para vocês entrarem lá no nosso grupo de WhatsApp, porque lá acho que é um bom lugar para a gente sempre manter vocês informados.
Segue o YouTube também, né? A página do YouTube da BrProt, que também
Isso.
vai ter. Todos os seminários estão depositados lá, né? Quem não assistiu de primeira pode assistir, né? Voltar no seminário e tá lá.
É isso. Beleza. Eu sinto saudade de encontrar vocês toda terça. A gente vai se falando.
Vamos manter contato, né?
Essa pandemia está me deixando emotivo.
É bom.
Gilberto,
É bom.
se cuida aí. Saudade de você. Beijo para todos.
Saudade de todos. Um beijo para todos.
Beijão em todos, pessoal.
Beijo,
Um abraço
tchau, tchau.
grande.
Cuida-se, por favor, que o time vai se reunir de novo daqui a pouco, hein?
É isso aí.
Isso.
Tchau, galera.
Tchau.
Tchau.